Okay. All right, so I have a few announcements / reminders before I get back into the cell chapter today. So the chapter 3 outline is due today. I forgot to remind you guys on Monday, but I won't remember to remind you guys for all the future upcoming chapter 3 outlines, or for all the future chapter outlines. So get that turned in if you haven't already. The idea is to get the outlines in kind of when I start the chapter. So it might seem a little weird, but at least you'll have seen the material before I talk about it up here. The disease selection spreadsheet is due today. So if your group hasn't submitted that, it's due by midnight tonight. I'll be going through those tomorrow and kind of assigning the different groups their diseases. So get that in if you haven't. Only one person needs to submit it from the group. It's not worth any points. So don't stress about it too much. And then the first exam is Monday. So the first exam is scheduled for Monday. I should finish up all this material today. So it's chapters 1, 2, and 3. The study guide's been up for a while to help you with your outlines. We'll do some reviewing on Friday. And I'll talk a little bit more about what the exam consists of, but it's pretty straightforward. If you've been in any of my classes before, it's going to be multiple choice and fill in the blank with a word bank. So just kind of a bigger multiple choice, I guess. You just have to write the word out. Yeah, word banks are good. I tried it without a word bank before and it did not go well. So we have word banks. So yeah, 45 multiple choice, 5 fill in the blanks with a word bank. And usually I put a couple of bonus questions on there as well. And I'll have like a little interactive quiz, either quizzes or Kahoot on Friday, and we'll do that and you'll kind of see the questions that I might ask. I guess that's it. And if we have to finish up any of this material, I'll do that Friday too. Any questions about what's coming up? Okay, so we're going to continue on in the cell structure chapter. I started with the membrane structure, so phospholipids, cholesterol, and then some glycolipids. Now we're going to talk specifically about proteins and how things move across the membrane, which is really important. Things need to get into and out of a cell. Membrane proteins do a lot of that. So like I said, if you count up the number of molecules on a cell membrane, most of them will be a phospholipid, these little guys, the head and the 2 tails. But by weight, these proteins, they don't make up a big number if you count up the molecules, and only 2% if you count up the molecules, but they're massive. So these huge proteins are stuck in the membrane. That's what, depending on the type of protein, allows certain materials into or out of the cell in a large way. If things can't move right across the membrane through the phospholipids, I'm not going to get into what does and doesn't necessarily. Most of the molecules that are moving into and out of a cell are going through these proteins in one way or another. So I'm going to talk about that now, I think that's what's coming up next. Yeah, membrane transport. Okay, so we call the plasma membrane selectively permeable. Permeable just means things can go through. Selectively means it's selective. It doesn't just allow any old thing to come through. You can kind of separate this movement into 2 major categories, passive and active. Passive mechanisms don't require any ATP or energy. So anytime you see ATP, that's just energy. The 3 kinds of this I'll get into briefly: filtration, diffusion, osmosis. Hopefully at least 2 of those terms sound familiar, diffusion and osmosis. Filtration, I don't think I cover that in general biology, but it's a very simple concept that I'll cover here. Maybe other professors do, but diffusion and osmosis, hopefully that sounds kind of familiar. Active mechanisms are anything that does require energy. So something that requires energy is going to be active. And then you have kind of different forms of active movement. Some of those are carrier mediated. So carrier mediated just means there's a protein carrier. So the protein is the carrier, it carries stuff from one side of the cell to the other. You can also have passive carrier mediated movement. So a protein might need energy to move stuff across the membrane, or it might not. So you have passive and active carrier mediated. We're mostly going to be focusing on carrier mediated mechanisms for the rest of this lecture. Okay, so that's the basics of membrane transport. Now I'm going to get into some of these subcategories here. First, filtration. We'll start with the passive stuff that doesn't require energy. Filtration is just due to physical pressure. So if you think of like your blood vessels, there's a lot of pressure pushing the blood and all of the substances in that blood through the blood vessels. So filtration, basically, that pressure pushes some of the particles out through the walls of the blood vessel. So water will move out. There's tiny little pores and breaks between the cells that make up your blood vessels. So that pressure will just force a little bit of solute or solution out of the blood vessels. That's all filtration is. So kind of squeezing material out of what I think of as kind of like a hose if you're thinking of blood vessels.
So for the filtration, diffusion, osmosis, are those the subcategories for the passive mechanism? Okay.
Yeah, so those are those are the three subcategories I'll talk about for passive. And then active, I'll give you an example basically. Yeah, so this is our first passive mechanism, doesn't require any energy, it's just due to pressure. Then we have simple diffusion. When I say simple diffusion, all I mean is where molecules are moving from where there's a lot of them to where there's not a lot of them. So down the concentration gradient is kind of the common term for that. So high concentration to low, that's diffusion. Molecules want to move from a high concentration of themselves to a low concentration of themselves. That's what they do naturally without energy. If the body needs to move particles in the opposite direction up the concentration gradient, that's where active mechanisms come in. You need energy to go against or up this concentration gradient. Simple diffusion, very simple. You have a lot of solute particles on this side of this semi-permeable membrane. Over time, they're going to even out. So there's an equal number on both sides. So they want to spread out. They want as much space between themselves as possible. That is simple diffusion. Osmosis is really closely related, but it is specifically dealing with water. So when you think osmosis, we're going to refer to it only as water moving through a semi-permeable membrane. There's a couple different ways you can think of how this moves. It is just simple diffusion. So a simple diffusion of water particles. So water, if you imagine water as kind of individual H2O molecules, it's moving from where there's a lot of them to where there's not a lot of them. So high water concentration to lower water concentration. That's what's driving the movement of these water molecules across a membrane. So if we look over here, the water molecules are these tiny little blue droplets, and then the orange is the solute. So there's basically more water on this side than there is on this side. The solute's taking up some space. Over time, the water will move to the other side to even out the concentration, and the water level will go up. Here we're talking about solute particles. So a salt, for example, that can't move across the membrane. So the salt, these little orange bubbles are stuck on the right side. They can't move, only water can move in this example. And an easier way how I think about it is water is moving towards the higher solute concentration. So water wants to dissolve that solute So it's going to move to where there is a higher solute concentration. That's going to come into play when we talk about hypotonic, hypertonic, and isotonic solutions in just a minute. So we're going to compare the relative solute concentration on either side of a membrane. Water is always going to move to where there's more solute. And this is really important for IV fluids. So this is where that story that the video I played for you guys on I think it was the second day of class about Emily Jerry. The pharmacist who created her IV solution put too much, basically there was way too much salt in it. There was too much solute, so water got sucked out of her cells, and she passed away. So it's all about water movement in relation to where the solute is. Osmotic imbalances, that's sort of what we refer to as if water isn't balanced on either side of a cell, can underlie diarrhea, constipation. Those would be kind of opposite directions, right? Or edema, which is swelling. So you want to know the term edema, that'll come back up. Swelling basically means a lot of fluid going into one area of the body. And there's a number of different causes of that, but for now, just know that osmotic imbalance is the cause, the ultimate cause for that. Okay. That is osmosis. Any question about which direction water moves? Okay, towards the solute. That's how I think of it. So now we're going to get into these terms, which hopefully you probably saw if you were making your outline. You probably have seen in a general biology class: hypo, hyper, and isotonic. So the tonicity is just the idea of relative solute concentrations on either side of a membrane. So if we're thinking about a cell, we'll think about a red blood cell here. We're talking about how much solute is there outside of the cell versus inside the cell. It's all relative to each other. So a hypotonic solution, and when I say hypotonic solution, what you want to imagine is, say, a red blood cell stuck into a beaker of water, and around it is the hypotonic solution. Hypotonic means there is not a lot of solute. Hypo means low. So if the solution is hypotonic, it has a low concentration of solutes. Tonic refers to the solute. So you can think of it as salt, NaCl, if you want to. That means if it's a hypotonic solution, the amount of solute inside the cell is a lot higher than outside the cell. So that's going to draw water in. So water is going to move towards the direction of the higher solute concentration, which would be into the cell, and that causes the cell to swell up. So this is where I think I mentioned ICF and ECF before. The intracellular fluid, ICF, is inside the cell. The ICF would be hypertonic, and I'll talk about that term on the next slide. So ICF would be hypertonic compared to the solution around it, which is hypotonic. You always have one and then the other. They're opposites. Questions so far? All right, if we look at hypertonic and then isotonic, isotonic's easy. Iso just means the same. Hypertonic would be the opposite. So if we're going all the way over here to this right side, now there's a whole lot of solute particles. So a lot of salt outside of that red blood cell. More solute outside the cell than inside if you have a hypertonic solution. So that's going to suck water out of the cell. Water is going to move towards where there's more solute, and the cell will shrink and shrivel down. An isotonic solution is what our red blood cells are in. That's where they're happy. So in our body, the concentration of solutes inside the red blood cells in our blood is the same as the concentration of solutes outside. That makes it isotonic. Iso means the same. So water is moving in and out, but it's an equal movement in and out. So you don't have shrinking or expansion. It just stays in that happy little indented donut shape. So normal saline, that's what goes into IVs, is 0.9% NaCl, so 0.9% solute. And that is basically the solute concentration of most of our body fluids. So putting that into our body isn't going to throw off the osmotic balance. It's not going to shrink our blood cells down or make them expand and potentially burst. So that's why getting the right amount of solute in a saline solution is really critical. Questions about any of those terms? It's all about water movement. Okay. Into that term I mentioned before, so that was all osmosis, more or less, just water movement. Now we're going to talk about carrier-mediated transport. This is where there's a carrier, a protein, that's needed to move something across a membrane. Before, all of that movement was just through the membrane itself. We didn't need a helper. So this is where we need a helper to get something that's really big or just won't cross that hydrophobic membrane, or it won't cross by it very well. So, sorry. 3 mechanisms of carrier-mediated transport. I deleted 1, so there's just 2 that you need to know. It says 3, I only listed 2. So we're just going to talk about 2. Facilitated diffusion is the passive, and then active transport is the active, obviously. So we talked about diffusion. Diffusion can happen with or without a protein helper. If you do need this protein helper, it's facilitated diffusion. So it gets facilitated by a protein, it gets helped. It's still just diffusion. Do I have a slide on this? No, okay. So I'm just going to describe this here without an additional slide. So diffusion is still just the solute moving from high concentration to low. It just needs a little tunnel, a protein tunnel, to make it through. But still no energy needed. It's just simple diffusion moving from high concentration to low concentration with the help of a protein. That's facilitated diffusion. Active transport always requires a protein. So you can't have active transport without some kind of helper protein making it happen. And now active transport, we're going to go against the concentration gradient. So from a low concentration of the solute to high, which is the opposite of what molecules will naturally do. That's why we need some energy. And that's what I'm going to go into next.
So if the primary active transport goes against the concentration gradient, I'm thinking that, did you just say that with the facilitated diffusion, the passive one is goes down the concentration?
Down or with, yeah, exactly. You'll hear down, you'll hear with. That's just two different descriptions of it. Yes, exactly.
So there's one that goes down with the concentration gradient, and then there's one that goes against it.
Exactly.
Okay.
Yep. Yeah, whichever of those terms makes more sense to you. So we're going to talk about now active transport, which goes against the concentration gradient. So we're moving molecules where they don't really want to go. And this has to happen in our bodies. Okay, so yes, I think I just said all of this on the previous slide. Active transport moves a solute through a membrane using a protein up or against its concentration gradient. You need energy for that. It doesn't happen naturally, so you got to kickstart it a little bit with some ATP. If you have an older version of this PowerPoint, I have a couple of different examples here. I'm only going to do this one just to simplify things. I think if you guys understand the sodium potassium pump, I'm happy. So I had primary and secondary active transport. We're just gonna lump it all together and talk about one. So don't get confused by that. I did change this a couple days ago. Okay, so the example, who remembers the sodium potassium pump? At least the term. Yeah, okay, good. So a few people remember it. It is so critical in the functioning of our body. So I definitely want to go over it now. You can already guess what's being pumped, sodium and potassium, right? And I'll talk about generally how that happens, not too much detail, but I'll talk about its significance as well. The idea behind it is that sodium is going to be kicked out of the cell, potassium is going to come in. When we get to muscle contraction, this is a huge part of what allows our muscles to contract and our nerves to fire. So the sodium potassium pump is really critical.
Oh, is that a new slide? I mean, is that was that one of the slides that you had, or is that the?
This one, or
Because this one says the sodium potassium pump. Did you put up new
that
slides?
one? Yeah. Yeah, I just said I changed them a little bit. So this should be, yeah, so this is just the only active, I'm not gonna talk about that, so cross that out. And then I changed a few words on here, but it's the same basic information. I cut it down just a little bit.
Okay.
Yeah. So here's sodium potassium pump. It's a protein carrier that is specifically dedicated to moving sodium out and potassium into the cell. The idea is keeping potassium concentration higher and sodium concentration lower in the cell, right? So we're trying to get sodium out and keep potassium in. That's going to allow for this whole, we'll unleash it basically to allow simple diffusion eventually to happen. That's what allows for muscle contraction. So I'm not even talking about, I know in general biology, whoever you had probably focused on 3 sodiums going out, 2 potassiums coming in. I don't even mind if you don't have to remember those specific numbers, but you are kicking out more positive charge than you're keeping in. So that's going to create this negative resting membrane potential. When we get into muscle stuff, I'll talk about negative membrane potential, resting membrane potential. You basically have a negative charge inside the cell because you have fewer positive charges inside versus outside. So all of our cells internally, when they're resting, are negative. So they're negatively charged. It's like a battery. So you have a negative charge inside the cell, positive outside the cell. That's what the sodium potassium pump does. It creates a negative internal charge. Without that negative internal charge, we can't use our muscles, any kind of muscle tissue. We can't use our nerves. Nothing happens. We need this charge difference across our cell membranes. Take my word for it. I won't get into more detail about it right now, but 50% of the calories that you ingest every day are used for sodium potassium pumps. That's insane. So half of what you eat basically goes towards creating this negative charge inside your 30 some odd trillion cells in your body. It's pretty amazing. So that's why I want to focus on that just briefly as kind of our one example of active transport. And again, it's active because there's already a lot of sodium out here, but the cell is kicking even more sodium out. So both of these are going against or up their concentration gradient. So sodium going out is against its concentration gradient, potassium coming in, also going against its concentration gradient. So it does require a good bit of energy to make that happen. Questions about that? I had something in the slides about sodium glucose co-transporters. We're not going to go into that, mostly because they don't really come up all that much anymore. They might later on, but sodium potassium pump I want you to be familiar with. That's the example of the active transport mechanism that I want you to know. Any questions about that before we kind of switch gears? Okay, so next is the organelles. You probably learned these in high school. You learned them again in general biology. You're probably really sick about us learning the organelles. So I decided rather than going through these slides, we're going to play bingo. We're going to play organelle bingo. So I have a couple of things that I'm going to hand out. And there's Starbursts for prizes for whoever gets the first few bingos. I have, for you guys on Zoom, I uploaded a document that says, I think, organelle bingo, and it has the word options and then the board on the back. You guys are going to create your own bingo board because I didn't want to create a bunch of different ones. So you get to choose. There are 16 words here at the bottom. You get a free space, so don't forget to put the free space in. You are going to take these words, these different organelles or parts of organelles, and put them in the 16 boxes on the back here, wherever you want. Totally up to you. It'll be fairly random. And then we'll go through some slides, and you'll have to figure out, based on the description that I have, what it might fit with on your board. So I'll start with that. I have printouts of these slides for you as well that I'll hand out. So you can use notes, I don't mind. This is just a slightly more fun way to learn organelles. Okay, so take like 3 or 4 minutes, fill in your board. Oh, does anyone want the handout, the slide printouts? Okay. If you don't want them, you don't have to take them, but it just has the description of what each organelle does. It's the rest of this PowerPoint, basically. Want that? Yeah. You want one? Yeah. Who else wanted them? Okay. So you can use your notes. This isn't like a quiz. And you can ask your friends if you want to. You can take back slides that are on the PowerPoint to have access to the PowerPoint. And this is the information you need to know about these organelles. Anything beyond this, you don't need to know. I'm just going to ask you questions based on what I have, or on the exam. I'll just ask you questions based on the info I have on these slides. So for you guys on Zoom, unfortunately, I can't send Starbucks either through Zoom. But if you guys want to play along, feel free. And like I said, the slides that are up here are just the organelle information. I just printed it out for people, but you can look through the slides on Canvas and you'll have the same info. Okay, and I think I'm going to stop sharing here. And hopefully you guys can see the screen. I'll read out the clues as well, but I think you can probably see that. Okay. I haven't done this before. We'll see how it works. I just get bored of talking about the organelles. You guys sure get tired of hearing about them. I'll blank on any of these. I didn't put these in any specific order. They're just kind of random, but I'm just gonna run through these slides. Each time you think you have an organelle written on there, that is described by the clue, just like cross it off somehow and write the clue number, just so we can kind of keep track of whether you're right or wrong. Yeah, you should have every single word on there. There's only 16 words on that bullet point list. I tried to think of more, but I got lazy. That's why there's not 5. Yeah, you can cross them off. Yeah, there's some additional ones, and not just the organelles. So. All right, take another minute or so, then we'll get started. So we can get finished in time.
Wait, so you want us to just, you want us to write like the clue that we got?
Yeah, so I have the clues numbered, so it'll be like clue 1, 2, 3. Just write 1, 2, or 3 in the box, and then like cross it off so you can keep track of when you get bingo. Yeah, usually bingo has 25 squares. I didn't want to put 25 words on there, so we have 16. I'll leave each clue up for about 30 seconds. I'll see if that is enough time for everyone to kind of figure out what the organelle or part of the organelle is. And yeah, just let me know the first person who gets bingo, you get some Starbursts. You get to pick your flavors. So if you like a specific flavor more, I think pink is the best. All right, are we ready? Missing one. All right, here we go. Clue 1, a double membrane that regulates molecular traffic into and out of the nucleus. Don't say what it is because other people have to, but no. Close, but no.
Oh my God.
All right, ready for the next one? Are you guys still searching? Good.
You said just like write number 1. Like
Yeah,
one.
just like cross out the square so you keep track of what, and then write number 1 in there somewhere, just in the corner. I don't care where. Just so I can double check and make sure. I don't, that probably doesn't matter, but just keep track. Okay, clue number 2. These are the monomers of a protein. If you guys on Zoom can't see that, let me know. It looks like you can. Ready? Next. Okay, I see some blank stares, but I'm gonna go on. Okay. Membrane-bound organelle that breaks down the cell's waste. All right, ready? Slightly tougher one. It's not quite as well known. Okay. Number 4, a membrane-bound structure that transports completed proteins to the cell membrane for export. That's a big one. I'll give you a little bit longer for that. Ready? Ready. Number 5. An organelle that directs most of the cell's activities. Ready? Okay. No bingo yet, huh? Number 6, a series of channels attached to the outside of the nuclear envelope with ribosomes attached to it. So the channels have ribosomes attached. Nobody yet? All right. Are we good? Okay. Next. A gel-like substance inside the cell that contains the cell's organelles. Yay. Okay. Awesome. Bingo. 1, 2, 3, 4. Boom. All right, we're going to keep going for a little bit longer. Oh.
I was just, I saw a C, got really excited.
All right, I feel like we're going to have like a waterfall whole. It's going to be title on this next one. You had to bring it back up. And you didn't even get it. Thank you. All right. keep going for a little bit. We have 10 minutes, 9 minutes. Membrane bound organelle that detoxifies substances. And specifically is found in abundance in the liver and kidney. Awesome. Nice. Anyone else? No? All right. All right, ready for number 9?
No, because you said membrane bound,
Yeah.
and I still got that, this is free space.
Oh, yeah. Yeah, they go. You got diagonal.
I had to look at it, and I was like, really?
All right, number 9.
Thank you.
You're welcome. The organelle on which amino acids are assembled into proteins. I'm talking specifically here. There's a couple of potential options, one of which you may have already marked off. So you got it right.
Oh, wait,
That's it.
Oh, bingo. I'm gonna go into everyone at bingo.
I just give some answers.
Yeah, well, the answers are, yeah. Yeah, we can go back and do the answers if we have time. I'm gonna put this on.
Bingo.
Sweet. All right. Next. No. This organelle adds finishing touches to proteins. You can splice them, you can add carbohydrates.
Bingo.
Yeah, pass those around. You need to still go a little bit further for more people to get bingo, right? Yeah. Do you want to keep going or do you want to go over the answers?
Well, I want to go over the answers because I think I did one wrong.
Okay, so we're gonna go back to 1 and start. All right, number 1.
Okay, bingo, wow.
Double membrane, what's the double membrane around the nucleus? Nuclear envelope, yep. All right, number 2, what are the monomers of proteins? Amino acids. Membrane bound organelle that breaks down the cell's waste. Lysosome, yeah. Who hasn't gotten starburst yet? Here. And then pass it to whoever else. Everyone needs starburst. Okay. So yeah, that's the lysosome. It's like the waste collection. Membrane bound structure that transports completed proteins to the cell membrane for export. Not really, not cytoskeleton. Huh? Yeah, the Golgi vesicles. So Golgi vesicles, it's funny, when I typed in Golgi vesicles into my phone for number 4 as the answer, it wanted to change it to vehicle. So that's a good way to remember. It's a vehicle to transport proteins to the cell membrane for export. So thank you, autocorrect. So Golgi vesicles. What organelle directs most of the cell's activities? Nucleus, yeah. So the nucleus is like the president of the cell. The president directs all of our activities. That would be not good. The autocratic leader, I don't know. Series of channels attached to the outside of the nuclear envelope with ribosomes attached. rough ER. So there's smooth and rough, that's endoplasmic reticulum, that's ER. They do slightly different things, but they're all kind of connected channels. Rough ER just has ribosomes studded on the outside of it, so it looks rough. Gel-like substance inside the cell that contains the cell's organelles. Cytoplasm, yeah. So that's just kind of the liquidy stuff, like Jello. Membrane-bound organelle that detoxify substances in the liver and kidneys. Yeah, peroxisomes. So your liver and your kidneys detoxify a bunch of stuff. They have a lot of peroxisomes. Number 9, organelle in which amino acids are assembled. Proteins. Ribosomes. Yeah, you could say rough ER too, because the rough ER has ribosomes on it. I would accept that, but the ribosome is what I was going for here. Ribosomes make proteins. When you think ribosome, think protein. And then this is the last one we did, the organelle that adds finishing touches to proteins. Yeah, the Golgi apparatus, Golgi, there's some other terms for it, the Golgi body. And then I think there's 5 more in here. If you guys want to look through them, I'm going to post this to Canvas. The answers are in the bottom in the note section down here. So if you want to know the answers to any of these, they'll be down here. You can look at the other 5 if you want to quiz yourself. And that is it. I'll see y'all Friday. We'll just do a review on Friday. So just review for the exam. And lab today, yeah. I'll see you in lab. I'll see you in lab if I have you in lab today.